All right, today we are diving deep into the microbial underworld. We're investigating two of the most notorious bacterial families out there, Staphylococci and Streptococci. You can think of them as the mob bosses of the microscopic world. They're living on us, all around us, and they are responsible for a massive crime wave of infections. So let's open up the case file.
So think about this for a second. What could possibly connect a painful skin boil, a really nasty bout of food poisoning, and a sore throat so bad you can barely swallow? On the surface, they seem completely random, right? But as we're about to find out, there's very often a common culprit pulling the strings behind the scenes.
And that's what leads us to our two prime suspects, the Staphylococci and the Streptococci. These guys belong to this powerful class of germs called gram-positive bacteria. And today, we're going to run a full investigation. We're going to figure out how they operate, how we tell them apart, and we'll look at the trail of diseases they leave in their wake.
Okay, so first, a quick profile on our subjects. We call them a gram-positive cocci. Cocci is just a fancy word meaning they're spherical, like little tiny balls. The gram-positive part, that's really key. It means they have this super thick outer wall made of something called peptidoglycan. In the lab, this thick wall soaks up a deep purple stain. And here's the twist. Most of the time, they're just harmless commensals, basically roommates living on our skin and in our noses, not causing any trouble at all, until they do. So how do we start telling these two families apart? Well, the first test is a game changer. It's called the catalase test. See, our immune cells actually use toxic hydrogen peroxide to attack bacteria. Staphylococci have this brilliant defense, an enzyme called catalase that just breaks it down into water and oxygen. And what does oxygen and liquid do? It bubbles. Streptococci? They don't have this enzyme. So the first clue couldn't be simpler. You add the peroxide, and if you see bubbles, you've got staph. No bubbles, it's strep. It's the perfect way to split the suspects.
So let's follow the evidence. The bubbles tell us we're dealing with a catalase-positive suspect, which puts us squarely in Staphylococcus territory. But this is a big family with a lot of members. To find our main person of interest, we need another test, one that's going to really narrow the field.
And that test looks for an enzyme called coagulase. Now, this is an absolutely incredible trick. This enzyme literally hijacks our own blood clotting proteins to build a shield of fibrin all around the bacterium. It's like building a little invisibility cloak to hide from our immune system. The ability to pull this off is what separates the kingpin Staphylococcus aureus from its less aggressive relatives, the coagulase-negative staph, like Staphylococcus epidermidis. So, just how common is this kingpin? Well, get this, about 20% of us, 1 in 5, are persistent carriers of S. aureus. For many of us, it's living completely harmlessly inside our nostrils right now, but that means this opportunistic pathogen is everywhere, just waiting. And being a carrier is a major risk factor if it gets an opportunity to invade.
And when Staph aureus decides to make a move, its rap sheet is incredibly long and varied. Some of its crimes are what we call intoxications. This is where it releases powerful exotoxins that do the damage, like in staphylococcal scalded skin syndrome, or SSS, which makes the skin blister, or the violent sickness of food poisoning. Other times, it's about invasive proliferation, where the bacteria themselves multiply inside our bodies, causing really serious stuff like bacteremia. That's bacteria in the bloodstream.
Let's zoom in on one of its most devastating weapons, the superantigen. The toxin behind toxic shock syndrome, TSST1, is the perfect example. See, a normal immune response is very specific and targeted, but a superantigen basically hotwires the whole system. It causes this massive, chaotic activation of our immune cells, which unleashes a dangerous cytokine storm, leading to a raging fever, shock, and organ failure.
I mean, just look at this table. It shows you just how well-armed a Staphylococcus really is. It has adhesins. These are special surface proteins called MSCRMs, which stands for microbial surface components recognizing adhesive matrix molecules that let it grip onto our tissues like Velcro. It has a whole toolkit for immune evasion. It has various exotoxins for different jobs. And it has enzymes like staphylokinase, which can actually dissolve blood clots to help it escape and spread.
Let's really dig into its getaway tactics. Our body's main defense strategy is a process called opsonophagocytosis. It sounds complicated, but it's basically just tag and eat. We tag bacteria with antibodies so our immune cells can find and destroy them. But S. aureus is a master escape artist. It has a capsule to hide, It uses something called protein A to grab our antibodies and face them the wrong way, making them useless. And it uses enzymes to just chop up any tags that do manage to stick. It is a master of disguise.
And of course, we can't talk about Staph aureus without mentioning its superbug form, MRSA. That's methicillin-resistant Staph aureus. This strain has picked up an extra piece of genetic code called the MEK-A gene. This gene produces a modified protein that our best antibiotics just can't bind to anymore, which makes them completely useless. This is a huge, huge problem in healthcare.
Okay, we've profiled the Staph kingpin. Now it's time to turn our attention to the other side of that catalase divide. We're going after the suspects who didn't make any bubbles, the catalase-negative crew, the Streptococci. They play by a different set of rules, and they have their own formidable reputation, so we're going to need a different set of tools to pin them down. So how do we sort out the members of the Strep family? Well, there are two classic lab methods. The first is checking for hemolysis, basically seeing what they do when you grow them on a plate full of red blood cells. Some, like S. pyogenes, cause beta hemolysis, which is a complete clearing of the red blood cells. Others, like S. pneumoniae, cause a greenish, partial clearing called alpha hemolysis. The second method is Lancefield grouping, which is like fingerprinting them based on specific sugars on their surface, sorting them into group A, group B, and so on. This slide really lays out the detective work perfectly. You start with a gram-positive coccus. Is it catalase negative? Yes. Okay. Step 2. What kind of hemolysis does it do? If it's beta hemolytic, you see if an antibiotic called bacitracin can kill it. If so, you've probably found your culprit, Streptococcus pyogenes. If it's alpha hemolytic, you use a different antibiotic, Optochin, to nail down Streptococcus pneumoniae. It's a really neat, logical process. So let's zoom in on the most infamous member of this whole family, Streptococcus pyogenes. You probably know it better as Group A strep, or GAS. This is that beta hemolytic bug, and it's responsible for a huge range of human diseases, from the really common to the truly terrifying. And its rap sheet is a long one. On the non-invasive side, you've got pharyngitis, that's strep throat, and skin infections like impetigo. But it can turn nasty and become invasive, causing things like necrotizing fasciitis, the so-called flesh-eating disease. And then there's this really bizarre and dangerous twist, post-infection complications, where weeks later, your own immune system gets confused and attacks your heart, causing rheumatic fever. Necrotizing fasciitis is the stuff of nightmares, for sure. It's this incredibly fast-spreading infection of the fascia, the lining around our muscles. The destruction is driven by a nasty cocktail of toxins called streptococcal pyogenic exotoxins, or SPS. Some of these are superantigens, just like we saw with staph, triggering a massive immune overreaction that destroys tissue. Now, before we close these case files, we have to acknowledge the wider network. Our two main suspects, staph and strep, they don't operate in a vacuum. There are other related gram-positive cocci out there that pose their own unique threats, each with their own specialty. Hey, remember that coagulase-negative accomplice we mentioned way back, Staphylococcus epidermidis? Well, while it's not as aggressive as S. aureus, its specialty is forming biofilms. These are basically slimy, fortress-like communities of bacteria that love to stick to things like catheters and prosthetic joints. Once they're inside that biofilm, they're incredibly resistant to antibiotics and our immune system, making these infections a nightmare to treat. Another major player you need to know about is Streptococcus pneumonia, also called pneumococcus. This is the bug behind most cases of community-acquired pneumonia. Its weapon of choice is a toxin called pneumolysin that literally punches holes in our cell membranes. But here's some really good news. The pneumococcal conjugate vaccine, or PCV, has been a massive success, cutting rates of severe disease in young kids by almost 80% in places where it's been widely used. And finally, we have the enterococci. Think of these as the tough guys from the gut. Their real danger lies in the fact that they are naturally resistant to a lot of antibiotics, and they're really good at picking up even more resistance genes. This is where we get superbugs like VRE, vancomycin-resistant enterococci, which are a major problem in hospitals. So let's wrap up this investigation. What are the key takeaways? First, the catalase test is that great dividing line between staph and strep. S. aureus is the coagulase-positive kingpin, armed to the teeth with toxins and immune evasion tricks. S. pyogenes is that versatile group A strep, causing everything from a sore throat to flesh-eating disease. And the main thing to remember is that this is a molecular battlefield, fought with weapons like adhesins, superantigens, and enzymes.
And all this leaves us with one final, and I think fascinating question. All these bacteria we talked about are commensals. They are our constant companions, living on us every single day. So what is it? What exactly is the trigger that tips that delicate balance and turns a harmless resident into a deadly invader? That's the central mystery. Thanks for joining me on this explainer.